Okay, I am now recording this meeting. All right, computational and storage requirements. So as I mentioned, if you're running something like a NovaSeq or a High-seq, you're going to generate a lot of reads. You're going to typically generate gigabytes to terabytes. So you're going to need a lot of hard drive space. This is something to plan for in your experiments because you will need a place to store these. A lot of people have now shifted to cloud storage. They're storing a lot of their sequencing on Amazon S3. That's also a very good option. Keep stuff off site, and you just basically pay for the space you use per year. But this is one example of just one family from a gene discovery study looking for rare variants causing skeletal dysplasia. And as we see here, if you were planning on doing some kind of sequencing work on, say, a MacBook Air, think again, because just this set of sequencing would fill up the storage on one. All right, data analysis requirements. So in-house, it's going to depend a lot on what you're looking at. So if you're doing microRNAs are short. And if you're mapping microRNAs against microRNA precursor sequences, so short reads against short templates, you're going to quickly discover that you don't need a whole lot of computing for that. You can do that on a Raspberry Pi, or, I mean, my Apple Watch could probably handle that. If you're going up to RNA-seq, where you're actually mapping transcripts against transcriptome, you're going to need probably a much more powerful computer. And if you're doing something even more intense, like whole genome bisulfite sequencing, where we're going to have both a lot of sequence and a lot of heavy-duty statistics with large matrices, that's where you're really going to need to start looking for a bigger computer, and probably start looking at either the Hoffman cluster or the cloud, because you're going to be doing a lot more than a standard computer can do easily. All right, so talking about sequencing studies, one of the first things I want to talk about is coverage. Coverage is an important element of study design for sequencing, and it can usually talk about a few different things. So basically with sequencing, a lane in a flow cell for a sequencer will only give you a pretty fixed number of reads. So for example, 50 million of them is the number I typically use, although depending on your sequencing platform, you may get more, you may get less. If you use a MiSeq, you're going to get longer reads, but fewer of them. If you're using a NovaSeq, you're going to get way more than 50 million reads per flow cell. So unfortunately, for sequencing studies, our funding is not limited, which means we can only do a certain amount of sequencing for any given study. So what this means is we need to kind of find a balance between how many reads we get per sample and how many samples we want to run. If I'm doing a gene discovery study and I sequence the hell out of somebody's genome and I blow all my sequencing money on that one person, then I have an N of 1, and Ns of 1 are really difficult to publish with. By comparison, if I do very low depth sequencing, so I do a gene discovery study and I decide I want to do 1,000 people, but I sequence them very low depth, my sequencing starts becoming unreliable and I don't get really anything to publish, and I probably don't find anything conclusive. So what do I mean when I talk about depth? It's essentially the number of times I sequence things or the number of times something has been sequenced. And again, it's a balancing act between the amount of data I want per subject and the number of subjects I want in a study. So this is just an example, variant discovery study. So normally I'd ask my class if I have any hardcore genetics people out there, but obviously I don't have a class in front of me right now. But I'll give you guys a minute or two. Can anybody tell me what's sort of interesting about this pedigree and what you kind of see here and what you expect? And just put it up in the chat to everyone with what you think on this. And I'll give everybody a minute or two to respond, and I'll have a little sip of water. All right. Anybody out there in Zoom land have an answer on what's kind of interesting about this pedigree? All right, so Marco knows this one. This is a fairly extensively inbred family. So anybody who likes Game of Thrones, this is kind of what you'd expect to see. Actually, not even from the Lannisters. This is more like the Targaryen family. And what we see here is mother and father are uncle and niece. Mother's mother and father are also uncle and niece, I believe. Father's mother and father are first cousins. The grandparents themselves are also pretty extensively inbred. So this is are probably also extensively inbred. And in fact, this family comes from a region of the world where uncle, niece, and first cousin marriages are culturally extremely common. So when you have inbreeding cases like this, it's not too surprising that you generate some unusual rare diseases. And in this case, these two boys have skeletal dysplasia. And in fact, what we see here is two children with skeletal dysplasia, two children without. So the ratio suggests dominant, but realistically, the inbreeding suggests probably recessive. In a different case, here we have a bit more of a common disease where we have two families, both families have the same disease. And in this case, we can just see the disease running throughout the family. This looks like it's probably recessive, but I can't say for sure. But how are we going to deal with these cases? So in the first case, we have a very small family. In fact, nobody really is known to have this particular disease except these two individuals. So we would probably want to sequence these individuals at very high depth, make sure we're not missing anything. Also sequence some other family members, make sure that we figure out which gene, we can figure out which markers are and are not looking like they're connected to the disease. In this case, because we have more people to work with, probably can do quite a few more individuals. And in fact, we want to do some unrelated individuals as well as close family members to get kind of a picture of where the variant is. And realistically, what we would do here is we'd probably do high throughput sequencing on a few individuals, find some candidate variants, and then do low throughput testing for cheap in the rest of the individuals, and just see how well that variant tracks with the disease or not. So there's a very good chance that your variant of interest is a single base out of over 3 billion. So the human genome is about 3.3 billion bases long. There's some chunks of it that we're still not very good at sequencing, or that we don't even necessarily know where they belong exactly. But you can expect a human genome to be about 3.3 billion bases. And a lot of genetic diseases, if you're studying them, are about 1 base in length. And keep in mind, people have 2 copies, so you're actually looking at 2 copies of a 3.3 billion genome base, so about 6.6 billion, of which, again, if you have a dominant disorder, 1 base change might be the entire cause. So as I mentioned before, a common strategy, although this strategy is becoming less common, is sequence a couple of individuals at high depth to see if you can find candidates, and then sequence other individuals to basically try and confirm or refute whether or not that candidate is causative. Now that sequencing is becoming so much cheaper, a lot of times people are skipping out on the low throughput sequencing and just doing high throughput sequencing on everybody in a family. So in this case, we're going to probably bias for number of reads because we want to make sure that we don't miss anything. The other thing is there's no such thing as a perfect sequencer right now. Every sequencer makes sequencing mistakes. So if I sequence a base 1 time, the first problem is humans typically have 2 copies of each gene. So if I sequence a region only once, then I've only picked up 1 copy and I missed an entire copy. If I sequence twice, there's a 50-50 chance that I've just sequenced the same copy twice and I'm still missing a copy. If I sequence a region 10 times, that probability goes down, and I've now more likely than not covered both alleles. But the other thing is we know that even Illumina sequencing, which tends to have a low error rate, has about 2% error. So if I sequence the same base 10, 15, 20 times, if one of those 2% errors shows up, that's perfectly fine because I know there's going to be 2% error. So if I get one base that disagrees and 18 or 19 reads that agree on what the base is, then I kind of know what it is. A slightly different example is quantification. So quantification studies, a lot of times you'll be doing this with ChIP-seq or with RNA-seq, where you're really trying to count how many times you pick up a given read. So ChIP-seq is looking at what binds to, say, a chromatin motif. RNA-seq, you're typically looking at transcripts from a cell and counting how many reads you get that tie back to a specific transcript. And the number of reads, and essentially what you're looking at is the number of reads mapping to a certain feature. So here we have condition A, and we have samples 1 through 6. So region 1 in condition A, we get very little reads. Condition A, region 2, we get a lot. And this is in the first group. In the second 3 samples, the trend is reversed, where we get a lot of binding to region 1 we get a lot of binding. So basically we get reversal here. Obviously for a normal sample, you would want more than N of 3 per condition. But for here, you can kind of see where it goes. And the nice thing with these is you're typically looking for specific things, or you're looking for things that are more common. So coverage in its simplest form talks about one of two things. If somebody talks about coverage for an entire genome, then typically what they're saying is how many times the average base was read, or in other words, the number of total map bases in your sample divided by the number of bases in your template region. People may also talk about coverage in terms of coverage at a specific base. And if they talk about coverage at a specific base, usually it's some base of interest, like something that's thought to be causative of a disease. And usually they'll say this base was covered 20 times. And when you hear them say that, they really just mean that that base was read 20 times. And obviously something that was read 20 times, you're much more confident in than something that was read maybe 5 times or 3 times. And if something was read 100 times, then you can be pretty confident that the right answer is probably reached. All right, and in terms of coverage, this is probably the most frequent issue that comes up. Quick show of hands, so people get ready on the raise hand button. Who out there either has done or plans to do RNA-seq on cell samples or on human samples or any other kind of living samples? Okay, quite a few. So RNA-seq is kind of interesting. If you take a typical human cell and you do RNA-seq on it, anybody know what the most common RNA is you're going to find? If you know it, feel free to just put it into the chat window. Okay, yeah, ribosomal RNA. Does anybody know approximately what proportion of a cell's total RNA is ribosomal RNA? All right, I got a 90%. The number that I typically hear is about 70, 75%, but it's going to depend very much on the cell. But typically a cell, there's going to be more ribosomal RNA than every other RNA combined. Well, or there'll be more ribosomal RNA than every messenger RNA combined. So this can be a little bit of a problem because anybody out there actually studying, aside from metagenomics people, anybody out there actually interested in human ribosomal RNA sequences? Yeah, I didn't think so. I got 2 people raising their hand. They're interested in human ribosomal RNA sequences. Okay, I don't usually get people interested in that. So yeah, most of the time ribosomal RNA sequences, if you're doing a gene expression study, they just get in the way. And that can be really annoying if you don't get rid of them. So there's a couple different ways of getting rid of ribosomal RNAs. One of them, I'll go over those a little bit later. But what you run into is this situation, is you have a few molecules that are, you have a lot of molecules that are relatively low abundance. You have a few molecules that are super abundant. So ribosomal RNA was just an example. But in just about every cell, you're going to find highly abundant beta-actin. So beta-actin is another good one. Beta-actin you'll see a ton of, and beta-actin is one of the main components of the cytoskeleton. So just about every cell makes a lot of beta-actin. Along with beta-actin, certain cell types will have other major components. So for example, if you take immature red blood cells, you'll find that immature red blood cells tend to have a ton of hemoglobin in them, not surprisingly. And in fact, I've actually managed to crash a computer program because somebody had me look up part of the RNA sequence in immature red blood cells. And just about their entire RNA-seq read library was beta hemoglobin reads. If you look in cartilage, you'll see that cartilage is a lot of collagen production. So you'll find like 5% of cartilage's messenger RNA is type 2 collagen. So you'll find a few super abundant molecules. You'll also find some super rare molecules. Can anybody out there tell me what's a frequent super rare RNA message molecule that you can expect to be looking for one day? And I don't mean a specific one, but more of a class of them. Yep, transcription factors. This is the most frequent one that I hear about, is how many reads do I want for my RNA-seq? And this is actually also, so I work at Zymo, I occasionally have to deal with tech support questions, and a lot of times the question will come up, how many reads, how much RNA do I want? How many reads do I want? And a lot of people like to think there's a one-size-fits-all answer for this, but there isn't. The reality is, if you're trying to sequence something like type 2 collagen in cartilage, you can get away with probably doing a few thousand reads and you'll get everything you ever need. If you're trying to look for a transcription factor, just about anywhere, transcription factors are always low abundance. Even a transcription factor that you know is present and important is typically going to be very low abundance. So as soon as I ask if they're looking for a transcription factor and they say, yes, I'm interested in transcription factor 2, then I say, okay, you're going to have to do a lot of reads to be able to pick it up. Because the more abundant a molecule is, the more reads you get from it. The rarer a molecule is, the fewer reads you get from it, therefore the more reads you have to do. At a fundamental level, sequencing is a sampling problem. So if you remember from, say, elementary school math, where the teacher put a bunch of black ping pong balls and a bunch of white ping pong balls into a jar, and then reached in there, and you had to kind of predict the chances of pulling out a given color ping pong ball. It's essentially that problem. But now imagine that your jar is much bigger. You have about 1 million ping pong balls in there. There's a bunch of different colors. And you're interested in the golden ping pong ball that is 1 in 1 million. So if you want to get, if you don't know how many golden ping pong balls in there, but you want to figure out their frequency, you're going to probably have to reach into that jar about 10 million times at least, just to get a rough idea. All right, so what's kind of the least you need to know about coverage? So coverage is the number of times on average that you read each base in your template. If somebody talks about deep coverage, they're usually talking about sequencing a region a lot or sequencing a given base a lot. This can be used either to detect rare changes at this base, where maybe you're only looking for it in a small portion of cells. It's also useful if you're worried about sequencing errors, because doing a lot of reads of one thing, you can try and accommodate for sequencing errors. And that would be my dog yelling. You can if you think you're going to get more than enough reads out of a single flow cell or out of a single lane, there's a technique called multiplexing. So again, if you're getting 50 million reads per lane, but you really only need 10 million, you can do what's called multiplexing, and you can basically read, say, 5 samples in that lane. Sometimes coverage is a little bit of a property of your data. So for example, if you're doing whole genome sequencing, if you notice that you cover a region about twice as much as you expect to, that's a sign that maybe you have a copy number variant. On the other hand, sometimes coverage is your data. So for example, if you're expecting uneven abundances, the number of times you read a given messenger RNA sequence or a given microRNA sequence or a given sequence in chip is really telling you what you're looking for in the first place, which is how much of that sequence was present in your final sample. All right, and then there's replicates and controls. So I'll go through this part, and I'm actually finding that in an online format, I tend to go through this material a lot more quickly because a lot of this class usually involves discussions with students and asking people a lot of questions and back and forth, and obviously that's not happening. So with replicates and controls, why do we need replicates? Well, number one, our data is always based on sampling, but more than that, biology is just messy. If you work with mice, you can typically control their genetics pretty well. So for example, black 6 mice are so inbred that they're practically all twins, or at least very close to it. That being said, black 6 mice and just about every other mouse still get, random diseases. They occasionally feel sick. You don't want to have an N of 1 because what happens if your mouse just happened to be sick that day? So biology is kind of messy. You get biological noise, which is just variation among what are essentially identical organisms that should be identical. You also start missing out on accuracy of measurement, so you don't know how accurate or inaccurate or reproducible your measurement is, and you lose sample size. I don't know of any statistics or confidence measures that work on an N of 1, but anybody out there ever tried to do using PowerPoint p-value test using only 3 samples per group? All right, I don't see anybody willing to admit to that, and that's fine. Okay, I have one person who basically says no. I've known a few people who've calculated p-values using Excel on N's of 3 per group. And the important lesson there is just because Excel will let you do it doesn't mean it's right or you should. There really isn't a good statistical test to tell you how reliable you are with an N of 3 per group, because an N of 3 per group is just not usually a good idea. So replicates here. There's 2 kinds of replicates that are important to think about in sequencing. There's technical replicates and biological replicates. A technical replicate is taking the same sample and resequencing it. Technical replicates are really useful if you have, say, a time gap in your experiment or something changes in your methodology, and you want to make sure the same sample is giving you the same data either at 2 different times or with 2 different library preps or even with 2 different methods. Biological replicates. A biological replicate is taking essentially the same biological condition in different individuals and running those. One thing that's worth note here, and this didn't really exist when I first started, but if you're doing single cell sequencing, you don't get to claim an N of 20,000 or whatever. You can't automatically consider cells to be biological, to be just a biological replicate with a huge N. So in this case, we have our black mice and our tan mice. And in our black mice, we're going to sequence this black mouse twice. So this is going to be a technical replicate. We're going to sequence this tan mouse twice. That'll be a technical replicate. And then we're also going to sequence this other black mouse and this other tan mouse. So we have biological and technical replicates. Nice thing with sequencing is Illumina and the other companies making sequencers have done a lot of work to make sure that their reproducibility is very good. So typically between runs on the same sample, you're going to get a very high degree of correlation. Something you sequence once is going to look the same the next time. the main time where you see a lot of variation is you'll see a lot of variability in a low abundance mRNA, or you'll see a low abundant, or something that gets sequenced low abundance, because the lower your coverage or the fewer or the rarer something is, the more it's susceptible to noise just by randomly gaining or losing a read. So to make inferences about the population, you need biological replicates. And this is where I talk about libraries. So just like my UCLA class, I'm going to kind of follow a rule of calling for a restroom or whatever other kind of breaks every 10 or so minutes, or I'm sorry, every for about 10 to 15 minutes on the hour. So I think before I get into libraries, this would probably be a good time for people to take a break. So I'll pick up the chat again at 10 o'clock on the dot. It's now 9:49 on my computer. So everybody, you got a 10-minute break for bio or stretching or whatever else you happen to want to do. Check emails. And I will pick up again in 10 minutes. All right, I'm back and it is 10 o'clock. There we go. All right, so talking a little bit about library prep now. All right, so with library prep, you're pretty much always going to do a couple of different steps. For short read sequences, you're always going to start off with a sample of DNA or RNA, which ultimately you're going to get some nucleic acid. You're going to fragment that nucleic acid if it's not already short. You're then going to ligate on adapters. And adapters serve a couple different purposes. So adapters are going to cause the DNA to bind to the flow cell. They're also going to give sites for starting the amplification, and they're just they'll give a place. They'll also potentially have barcodes on them for identifying where the read came from. And actually, since I'm doing this virtual, so one of the things I can't do here is I can't draw on the whiteboard, but Illumina has some very good movies on this. Let me pull up one. All right, are people seeing my slides, or are people seeing the web browser in front of them? Ah, okay. So give me just a moment. All right, now people should be seeing a web browser screen, although this is just a YouTube video. All right, are people seeing the movie now? Okay, perfect. So this is actually going to show you how sequencing happens on a flow cell. Are people hearing the audio from the movie as well? Okay, so you're not hearing the audio. Okay, so I know so I can talk you through it. Okay, this is all intro. All right, so they're going to go over library prep now. Okay, so pretty much with all Illumina sequencing, you're going to add these adapters on the ends of your DNA fragments. All right, so the adapters are going to have a few key components. They're going to have sites here for interacting with your DNA sequence and starting amplification. They can have barcode sequences, And at the far end, they're going to have sequence that'll bind down to the flow cell. So the flow cell itself has complementary DNA sequences covalently bound to it. They can act as primers, and you'll see how that works in a moment. So this is a flow cell. All right, so inside of a lane in a flow cell, you're going to have this lawn of oligos that are covalently bonded to it. It really does just kind of look like a lawn of DNA sticking out. So you have your adapterized fragment, and it's going to hybridize down to one of the oligos in the lawn. And that oligo is going to become a primer. And you can use that oligo to then copy that piece of DNA that hybridized down. So you now have a piece of DNA that's essentially covalently bonded down to your flow cell. Once you denature it, the original template just gets washed away. And then you have something going on that's called solid phase PCR or bridge amplification, where you let the other side hybridize down, send in a polymerase, and synthesize a complementary strand. And this will now undergo solid phase PCR. And this is a process called colony formation, because this is actually going to grow out like a bacterial colony and form a colony of a single origin sequence on the flow cell. So because we have different oligos for the forward and reverse strands, you can actually cleave off the reverse strands so that you only sequence in one direction. So at that point, you're then washing over different fluorophores or fluorophore-bound NTPs. And at this point, you're just doing synthesis, and you're looking at the fluorescent signal generated with each step. So that process they show of it fluorescing and going away, that's not actually that instant. What'll happen is you'll add a fluorescent nucleotide. You'll read which color shows up for a given cluster, and then you cleave off the fluorophore. So it'll bind, it'll get excited by some kind of light to be read, and then it'll be cut off. And this is Illumina sequencing by synthesis in its essence. So this is showing you all the different clusters and how they're lighting up as nucleotides are being added to them. What's really cool about this is they actually, in order to detect where clusters are and identify a working cluster, they use the same software that the JPL and NASA use to identify new planets and new stars. So after it finishes reading in the first direction, they can wash away all the product from the first read, and then they can try and read in the reverse direction. So at this point, they're reading the index or the barcode. So the way that Illumina reads go is if you have a forward read, you're always going to read the forward insert first, then you're going to read the barcode in the forward direction afterwards. if you're doing reverse reads as well, then your first read will still be forward direction, your second read will be forward barcode, your third read will be reverse barcode, and your fourth read will be reverse sequence. So after you read the barcode read, you wash away that product. So failure to completely wash away the original product, plus the fact that you now are basically reading this bridge product, are why read 2s tend to always be worse than read 1s. So again, you've made that bridge product and you're going to cleave away your original read. Failure to cleave away that original read is one of the reasons why your reverse reads are never as good as your forwards. So ultimately, this is happening millions of times over the flow cell. And depending on the barcode patterns, you can do multiplexing. And I'll go into how that works in a bit. All right, and that's everything everybody needs to see from this. All right, give me 1 second. I'm going to get back my okay, so back to slides. Okay, and I got my chat window back. Does everybody feel a little bit more confident in their understanding of how Illumina sequencing actually works at the nuts and bolts level? Okay, cool. So now we kind of have an understanding of what the adapters are there for. We can do site. So after we ligate on the adapters, which all the reads need, we can then do a size selection. And size selection is basically making sure that we have the right insert size. We've gotten pretty good with techniques like sonication. So sonication is a really popular one for breaking up DNA sequence because it seems to be the least biased. But some people also need either need or want to use chemical methods for fragmenting their DNA, and that's also perfectly fine. But you want to make sure that you have consistent fragment sizes. If you have inconsistent fragment sizes, a lot of times your sequencing won't go well. And there's a lot of things you won't be able to say. So after adapterization, you basically, once this happens, you've kind of made your library. So at that point, you have to go to quality control. Nobody wants to run a bad library over their sequencer. It just wastes flow cells and money. So there'll be some kind of quality control steps, including things like quantitative PCR, looking at size distribution of the inserts, that sort of thing. And then finally, sequencing of your library. Side note, this quantitative PCR method that you see here is actually also the predominant thing people are doing right now for coronavirus testing. All right, so there's a couple of questions that you'll need to ask about the kind of libraries you're running. One of the important questions, are you going to do a single sample per lane or multiplexed? Especially now with things like the NovaSeq, you get way more reads per lane than you are going to need for most samples. So a lot of times you want to multiplex. And multiplexing is just using those index reads to identify the sample of origin so that you can have 5, 10, or more different samples on the same flow cell. Stranded versus unstranded. So this is also a very important question to check. A stranded library is not going to tell you strand of origin. I'm sorry, a stranded library will tell you strand of origin, and an unstranded library won't tell you strand of origin. If I'm sequencing, say, a human looking for a rare genetic disorder, I'm not really worried about strand of origin. And the reason I'm not worried about strand of origin is somebody who has an A in one position is going to have a T on the opposite strand, and that's pretty much going to happen everywhere. DNA mismatches don't really persist in cells. If I'm doing an RNA-seq library, I'm much more concerned about strand of origin. And the reason for that is, and again, you don't see a lot of this in humans, but you can have anti-parallel genes where you have the same, you have essentially the same stretch of sequence, but in one direction it's one gene, and the other direction is a different gene. Humans don't do a whole lot of this. It's relatively uncommon to find a gene in to find two genes that overlap in humans. Microbes have a little bit more common because microbes tend to be much more efficient with their genomes. And viruses, which are probably about the most genomically efficient things around, viruses like to do these overlapping in anti-parallel genes all the time. So if you're looking at expression of viral genes, you almost certainly want to do a stranded library. And then poly-A or ribodepletion. So fundamentally, when you're, as I sort of touched on before, in just about any cell you deal with, ribosomal RNA is going to be the most prevalent RNA in there. And that can be a major annoyance. If you're looking at differential gene expression, ribosomal RNA is usually not something that people consider a gene of interest for differential expression studies. So you just want to get it out of there. Otherwise, it's going to take up more than half your reads. There's essentially two ways of getting rid of the ribosomal RNA. One of them is direct removal of ribosomal RNA, and there's a few different techniques for that. The other one is to grab messenger RNAs by their poly-A tails and pull them out of the mix that way. So one option is you rescue the poly-A from, you basically rescue the poly-A from the sea of ribosomal RNA. The other option is you drain out the sea of ribosomal RNA and just leave behind the non-ribosomal RNA. Both these methods have advantages and disadvantages. So poly-A pull down kind of has the advantage that it tends to be cheaper, and there's certain processes where you can kind of automate it or make it or lower your input with it, which is nice. Poly-A pull down doesn't get a lot of things though. So for example, a non-coding RNAs don't always have poly-A tails, which means you don't get to grab them. So you lose non-coding RNAs. MicroRNAs never have poly-A tails. The other problem you'll run into is you're essentially grabbing a messenger RNA by its tail, which means if that messenger RNA breaks somewhere, you're going to lose its head. That's not as huge a problem if you're dealing with short sequences, but as an example, I was doing a study where I was looking at a gene that was about 15 kb long for transcript, which is an incredibly long transcript to be dealing with. And when I was dealing with that gene, what we discovered was we had huge biases because we did poly-A pull down, where we could very efficiently sequence the tail portion, but most of the time we didn't see the head. So most of the time what we were seeing was evidence that the messenger RNA broke somewhere during extraction, which isn't too surprising because it's 15 kb long. And this basically means that during extraction, because it broke somewhere, we got almost no coverage on the head, and we were interested in looking at sequence variations of the head. So that's another problem, is you'll tend to bias yourself towards the 3' end of genes and lose the 5' ends. So I said before, stranded versus unstranded libraries, you basically can figure out what the whether you're reading from a positive strand or a negative strand. All right, paired end versus single end. This is also another important question to consider. So as I showed in that Illumina illustration from before, paired end, you're reading from both sides of your insert. From single end, you're only reading from one side. So question here comes up, is getting more sequence always giving you more information? Or in other words, if you're getting 50 million reads per flow cell and you do paired end, is 2 times 50 million 100 million. So you will get more information for certain things. If you're looking for splice variants, you absolutely need to do paired end, because for picking it up, in order to detect a splice variant in single end, you would need to actually have that splice variant be somewhere roughly in the middle of your sequence. If you do paired end, as long as you have somewhere roughly in the middle of your insert somewhere a splice junction, you can kind of get an idea for what's going on. So for example, if I have a bunch of sequences on the same insert where the first paired end is giving me exon 1 and the second paired end is giving me exon 15, I've probably skipped a few exons in there. If I just do forward reads, I wouldn't be able to tell that because I would just have, say, exon 1. You can pick up structural variations this way, so things like genomic inversions, that kind of thing. You will get more coverage. So if you're just doing variant discovery, this is actually a very good option because variant discovery tends to be dependent on having a lot of read. You get better mapping, although that's usually not a good enough reason to convince me to do paired end, because realistically you only bump your mapping up about 10%, about 5% typically, maybe 10% at the most. If you're doing quantification, it's less interesting. So let's say I'm doing RNA-seq. If my forward read aligns to my favorite gene and my reverse read aligns to my favorite gene on the same insert, how many times did I see my favorite gene there? Quick show of hands, who says I saw it twice? Who says I saw it once? Yeah, so I'm seeing everybody raise their hand on this one. Yeah, if the forward read says I saw my favorite gene and the reverse read says I saw my favorite gene, then I still only saw it once. Likewise with something like ChIP-seq, if I see my favorite region in the forward and reverse read, I still only saw my favorite region once. All right, as far as multiplexing goes, so this is actually what multiplexing is and what's involved. So with multiplexing, you're going to start off with a couple different samples. So in this case, 2 biological replicates of black mouse and only 1 biological replicate of tan mouse. You're going to still generate your sequencing library just like before, but now you're going to see things where you have, for example, this one that just has kind of your standard adapters. This one, you have an adapter with a slightly different sequence here. And here you have an adapter with even a different sequence. So that when you do your sequencing, you read across this index region. And this will all be sequenced in one lane. And what will happen is when you do your demultiplexing, you essentially reverse this process and say, based on which adapter I see here, can I tie it back to a specific sample? So how much multiplexing can you do? That's actually a pretty simple formula. Once you figure out about how many, well, you're going to start off knowing how many reads you get per lane or whatever other unit of sequencing you get. You're also going to have a rough idea of how many reads you need per sample, and that'll tell you how much multiplexing you can do. So the rough formula or rule of thumb is your maximum multiplexing is the number of reads you get per unit or per lane divided by the number of reads you need per sample. As a general rule for this though, if this number comes out to something on the order of dozens, knock off about 10%. If you get 10% more reads than you need per sample, that's not a terrible position to be in. But occasionally flow cells underperform. So maybe a flow cell only gives you 35 million reads or 40 million reads. If you really maxed out on the multiplexing, you might find yourself in a position where you didn't get as many reads as you needed per sample. If your Nmax number here comes out to some single digit number, or maybe something like 10, knock off 1 or 2 of the samples. And again, just get a little bit of extra coverage in your existing samples. All right, so what's kind of the least you need to know here? Well, you want to make sure you know that biological replicates let you see variations within the same sample group. Again, biology is noisy. There's not much we can do to kind of get rid of that issue. Cells in a dish tend to not be as noisy a lot of times. Mice in, genetically identical mice in cages tend to be more noisy than cells, but they still tend to be pretty clean. Humans, free range wild type humans tons of variation. So doing technical replicates is important for identifying potential technical issues. And again, I highly recommend it if you do things like have your samples run at different times or different methods or anything like that. During library prep, you ligate on adapter sequences, and you may do some other things. So you may remove ribosomal RNA, you may do exon capture if you're doing exome sequencing. If you, do I have a message from, oh, okay. You may also remove, you may also capture mRNAs, or you may do CHIP or anything like that as kind of part of your library prep. CHIP, most people don't consider part of library prep, whereas either removing RNA or capturing mRNA or capturing exons, people do consider part of library prep in general. You may need to do stranded, although that depends on your application. And chances are you'll probably be doing multiplexed. A lot of times if you're doing multiplex, your core will do the multiplexing for you because they might even multiplex you with other people's samples. So we'll go through a little sample here. We're comparing the black and tan mice. So we have this black mouse that's generating samples 1 and 2, and that'll be a nice technical replicate. And so otherwise we'll get samples 1, 3, and 4 from black mice. We'll get samples 5, 6, and 7 from our tan mice. So we have 3 mice per condition. We have 1 technical replicate. We have 2 conditions, so it's a total of 7 samples. Then we have to ask, what information do we want to get? And as far as information we want to get, we might want to look at differential gene expression. That's a good single edit application. If we want to look at alternative splicing, that's going to definitely be paired end. Structural variants will be paired end. Genomic variation, that's probably going to be paired end. So we'll figure out how many reads we actually need. Then we can figure out multiplexing options. So let's say we're doing, so we're looking here at cost versus number of reads generated. If we do 7 samples per lane, we're going to get about 40, we're going to get somewhere in the neighborhood of 50 million raw reads. We figure we need 2 lanes, so we'll probably need about $2,000 to $4,000. This number's actually gone down quite a bit. So let's look at how we can set up these samples. For design 1, we're just going to run all of our black mice, including the technical replicates, in 1 lane. We'll run all of the tan mice in another lane. This is a bad option because you can't tell the difference between lane to lane variation and group to group variation. They're going to look identical. For design 2, we're going to have our technical replicate between the 2, so that's a very good way of doing things. We're going to have 2 of our tan mice in library 1, 1 of our tan mice in library 2, and we're also going to split up our black mice. This isn't a terrible way of doing things. The probably best way, especially if you're using a core, and they tend to do this a lot, is you just run every sample in every flow cell on the plate. So the nice thing about this is you have everything being run in both lanes. So you're still going to get the same number of sequences in the end. Any variation lane to lane is basically going to fall out because everything's been run in every lane. You can still compare these 2 for technical replicate purposes. But another nice feature of this method is there's no rule saying that you have to run this lane and this lane on the same day. So for example, let's say that I run library 1, and what I discovered was I underestimated the effect size of whatever it is I'm looking at. And it turns out with just half the samples run, I'm already looking at a hugely significant result, something that's very statistically clear for difference. And I kind of say, well, is running library 2 going to show me anything I didn't know before? And maybe there's some other genes I'm interested in which case library 2 might be worth doing. But if at library 1, I've proven everything I want to prove, and I've proven it pretty conclusively, then maybe I don't even bother running library 2. That's probably $1,000 or so that the lab doesn't have to pay. So put it towards beer money or something good like that. So what are you going to get out when the sequencing is all said and done? You're going to get a bunch of these gzip fastq files. And here I'm actually showing QSeq. Does anybody out there ever get QSeq samples or samples back in QSeq format? Raise your hand if you get back QSeq. All right, raise a hand if you get back FastQ. Okay, that's kind of what I was guessing. I think there's only one core on campus that really likes giving people back QSeq. They're kind of the odd ones on that one. Oh, okay. BSCRC isn't doing QSeq anymore. They're doing QSeq and FastQ according to Marco. Okay, well, it took them some time to get around, but at least they're getting around. Basically, a long time ago in a galaxy far away, actually no, a long time ago in a lab that is nearby, there was a war between two different formats for sequencing data, QSeq and FastQ. FastQ won. FastQ hands down won. Most sequencing applications now expect FastQ data. I don't know of any sequencing applications that expect QSeq. That being said, the Broad stem cell sequencing core, they still like QSeq for certain pipelines, so they'll return you data in QSeq format. Which everybody who gets QSeq, the first thing they ever do with it is they immediately convert it to FastQ. Although it sounds like now they're giving people FastQs for something, so there is still hope. Either way, there's got to be a lot of data here because there's a lot of sequence, so you're going to need a bigger hard drive. Also important to note, make sure you pick the right application for your question. If you're looking for genetic variation, do either whole genome DNA-seq or do exome-seq. If you're looking to quantify things like gene expression or methylation or that kind of thing, if that's your main goal, do RNA-seq, do your microRNA-seq, do your ChIP-seq, do your bisulfite-seq. I know of some people who try and use, who've tried to use nanopore for bisulfite-seq, because nanopores can theoretically, with some not so good amount of inaccuracy, detect methylated DNA. Realistically, it winds up being inaccurate, and I've never seen anybody get anything conclusive out of it. I've seen a lot of people, and actually worked with a lot of people who were doing RNA-seq on tumors. And they were doing this look at gene expression in the tumor. And then after they finished their gene expression in the tumor, they said, oh, RNA-seq should be theoretically like a exome sequence. So let's run the RNA-seq and see if we can get a genomic sequence out of it as well, and see if we can find some new genomic variants. It sounds like it should work in theory. I have actually seen this fail so many times. I've never seen it work. The only time I've seen successful genomic variant detection by RNA-seq is from a group that had very limited numbers of variants they were interested in. Specifically, they wanted to find some p53 mutants, which that was easy enough to do because I could just go through the p53 sequence and see if I could detect them. But even there, a lot of times with the p53 variants, I said, look, you didn't have enough coverage of this p53 region, so I couldn't make a confident call about variants. So if you want to find genomic variants, do your genomic sequencing. If you want to find expression variants, do your RNA sequencing. Don't assume that you can get something for free out of it. It turns out trying to get genomic variants out of RNA-seq takes about 10 times the amount of bioinformatic effort, and the results are usually pretty much unusable at the end anyway. So try and avoid doing this operation where you try and go from these quantitative expression studies to finding genomic variation. All right, so what do you need to know about this part? Essentially, make sure that you're using the right tools and the right application for your job. Make sure you plan the experiment carefully. If you're not super versed on sequencing, maybe run through the experiment with somebody who is, who can give you some ideas if you're getting enough coverage and if you have it well planned. Obviously, you need to obtain samples. Make sure you're getting the right kind of samples. If you're doing library prep, Make sure you know how to do library prep well. If you're not used to doing library prep and you don't plan on doing library prep much in the future, consider just paying for the core to do it. It's usually not that expensive. And unless you want to specifically learn how to do library prep or need to, the core is actually a very good option, because the core, since they do library prep day in and day out, their library prep techniques tend to be very reproducible, and they tend to have very low rate of failure. So run your sequencing, do your demultiplexing, and then do your analysis. Okay, and this is where I do an intro to Hoffman 2 and the UNIX command line. And I'm actually way ahead here, so let's do another break until 10:45, just for stretching, restroom, water, whatever. Make sure you stay hydrated, that kind of thing. And I will pick this up again at 10:45. All right, I think we can get started again. It's now 10:46, so yeah, we should definitely be able to get started. All right, so introduction to Hoffman 2 and the Unix command line. So this will be especially useful for people who don't have a lot of experience on the command line operation. All right, Hoffman 2 is what's called a computational cluster. So the idea behind Hoffman 2 is it's not actually just one computer. There's several hundred computers in there. And those computers are what's called headless, so they don't have keyboards, they don't have monitors. You only interact with them remotely. So you basically get a remote connection in there and you issue them commands over the command line. The amount of storage space and computational resources you get on there depends on if either your PI or department has bought special resources. But just by having an account on there, you get a certain amount of computational resource no matter what. So the max job run is still 24 hours, that's for sure. I've noticed they've let up on a lot of these other restrictions, like you get more RAM than 4 gigs now. You can request more processors. You still get 20 gigs in your home folder, and the stuff in your home folder stays there. It doesn't get deleted randomly. You also get 2 terabytes of space in your Scratch folder. However, the contents of your Scratch folder are subject to deletion after they sat there for about a week or two, unless they've been used recently. All right, so a quick refresher on the Unix command line for people. Basic computer architecture is you have what's called the kernel. You've probably heard people talk about kernels before. That's just essentially kind of the center of your operating system or the center of your CPU. This is where all the decisions are being made, where all the stuff is actually being analyzed. Your kernel connects to a bunch of devices on the outside, so it can connect to, say, a device like a USB hard drive. It can connect to a printer. It can connect to and open files, potentially from devices. It also takes in data from applications and runs applications. And it handles data coming in and going out on the network. So around the kernel, you have what's called a shell. And a shell is your command prompt. A shell is where your computer says, type a command for me and I'll do a thing. So you type the thing you want your computer to do into the shell. Once you type it in and hit enter, most of you are going to be running bash shells. So the bash shell will interpret it into machine readable instructions, send it to the kernel. The kernel is going to figure out what to do with it and see if it can do it. A lot of you for your personal computers are going to have what's called the graphical user interface or GUI wrapper. And that actually goes around everything else. And that can either generate commands over the shell to run, or it can pass directions directly to the kernel. But the GUI wrapper essentially translates computer commands into mouse clicks and movement and double clicks and drag and drops and all that other fun stuff. So computers didn't always have these GUI wrappers. I think Mac was essentially the first thing that really made it standard. Before that, especially on things like the early Apple computers and the early IBM computers, it was all just interaction with shell. The way that the kernel kind of decides what to do is you have your command prompt and you then have a $sign, and the $sign is kind of a sign that you put in a command. So your command is going to be do the thing, which might be running a Python file, might be running a Python script, or it might be reading a file or something. You usually have some options for it to do, so maybe some arguments or at least passing it a file name. That goes in through the shell, that gets passed by the shell to the kernel. The kernel is going to say, what are they asking me to do? And they're saying, okay, you're asking me to do the thing, I know how to do that. You're asking me to do it with the file, okay. And you're telling me how to do it. So if the kernel gets all those instructions and they seem correct, the kernel's then going to ask if you have privileges to do the thing. So it's going to ask, can you run this file? Can you read this file? Can you run this file? And if you can do all those things, then the kernel is going to say, okay, they gave me instructions I understand to do stuff with files that they have permission to do stuff with. So it's going to do the thing. Alternatively, if the kernel doesn't understand your instructions, you'll get back an error message, frequently because of a typo. Alternatively, if it checks to see if you have privileges to do the thing, if you maybe don't have permission to read one of the files you're trying to read, it'll return you an error message saying probably permission error. Here's some really common, really helpful command line commands that you'll encounter. So pwd will have it display your current working directory. The cd command will have it change directory, so you can change to another working directory. cd ... So this is a change directory command. The ... is Linux shorthand or Unix shorthand to say up a folder. So if you think of the Linux file system, it's basically a tree. It starts with a folder called / or root. And from there, it just kind of branches out. So if you do cd space ... you're moving one level towards that root folder. Less is a command to open a file in a text viewer. So if you do less and then pass it a file name, it'll open that file in text viewer. CP tells it to copy a file from where to where. RM tells it to delete a file. rmdir tells it to delete a whole directory. That directory has to be empty because rmdir won't remove the files. SCP is a command for secure copy, and that'll copy data over SSH from one computer to another. You're not going to use SCP command much in this class because I'm going to have everybody do this using an SFTP client. gzip will compress a file. There's also other zip programs that will compress files. gzip does uncompression. mkdir tells it to make a folder. So just like ... is shorthand for the folder directly above, is shorthand for the current folder. The tilde is shorthand for your home folder. ls, and you really don't need this asterisk here for it, but ls will list all the files in a folder. And wc will do word and line counts on files. So just a couple of useful commands. On the Hoffman cluster, software is located at /u/local/apps. And if you ls in that folder, you'll get a list of all the available software. Usually for each piece of software, you'll find there's multiple versions of it available. If you want to use software that's not available on the cluster, you can typically run the executable from your home directory. If for some reason you need administrative rights, you have to talk to the cluster administrators about it. Generally, they're not going to want to install something that'll require administrator rights, but you can always ask. A lot of times, if you try and do that, they'll actually show you a way to do it without needing administrator privileges. All right, so logging onto the Hoffman cluster. Quick show of hands, who's already logged onto the Hoffman cluster sometime in the last couple months? Okay, cool. So everybody who's logged onto the Hoffman cluster recently, you basically don't have homework tonight. Everybody who hasn't logged onto the Hoffman cluster recently, you'll have a homework assignment tonight, which is log into the Hoffman cluster via SSH, and just make sure that you can actually log in there. I have so many students who've had issues like they've forgotten their passwords or their account's been deactivated or stuff like that, where ultimately I asked them, did you try logging in? They said, well, I logged in a year ago and I didn't bother trying to log in recently. I was like, well, you can't really participate you can't really take part in the class exercises until you log into the cluster. So you're kind of stuck until the cluster administrators reset your password or do whatever they have to do to reactivate your account. And that sometimes has a turnaround time of the day. So on Mac computers, you're just going to open up your terminal window. The easiest way to get a terminal in Mac is press Command space, so the Apple key plus space. Start typing in command, and you'll see it come up fairly quickly. You just hit enter, and it'll come up with a command window. Windows, this has changed a little bit now that Windows is sort of integrating itself a little bit more with Linux, but it used to be you had to use something like PuTTY, or my personal favorite is actually MOBA Xterm. But you have to use some kind of a terminal program because Windows doesn't really have the same kind of terminal. They've had a lot more support now, so you can SSH from the Windows terminal if you really want to. Although Windows terminal is still not as good, so I tend to prefer MOBA Xterm. The computer programs you're going to be running are written in a few different languages. So programs written in C++, the source code ends with CPP. C++ needs to be interpreted or needs to be compiled into a machine runnable program. And once it's compiled, you can't really read it anymore. Java language, also compiled, runs in a virtual machine called the Java virtual machine. The idea behind this was that somebody just has to compile it once and it will run in every Java virtual machine. Realistically, from what I've seen, that doesn't seem to be completely true. There's also Perl language, P-E-R-L. This one's mostly fallen out of favor for bioinformatics, but probably about 5 years ago, it was still kind of the standard bioinformatics scripting language. Python is now very much the standard, and these scripts land with PY. So both Perl and Python are interpretive scripting languages, which means that you fire off a script and it actually runs on the fly through an interpreter that turns the commands into computer language. Because Python is caught on so much more strongly, it's gotten a lot of optimizations, and in fact, Python tends to be very fast even for an interpreted language, although interpreted languages tend to be slightly slower. If Python's something you want to learn, feel free to shoot a message to the collaboratory. We have a lot of Python training available. So when you log on to the Hoffman cluster, you're going to see a few things. It'll ask you for your password, and this is very important to note. As you type your password in here, you'll see nothing happen. You won't see asterisks appear, you won't see dots appear, you won't see a cursor move. It's just sending what you've typed in for your password to the host computer and not displaying anything, and that's kind of a security thing. The backspace key also does not work when you type in your password for command prompt. So if you know that you made a typo in the password, the best thing to do is actually just hit the enter key, fire off the wrong password, and then type it more carefully the next time. A few other things you'll see. You'll see a statement there telling you not to compute on login nodes. And that's because everybody who's connected to the Hoffman cluster is connected through a login node. So if you compute on that login node, everybody else who's logged in through that node, even if they have an interactive session, they'll either get booted off because you've taken up all the resources by doing computing on a login node, or those sessions will become really slow, and you may get your job terminated, or you may get and you are likely to get an nasty note from the administrators telling you not to compute on login nodes, and they'll kick you off if you keep doing it. It really does kind of crap things up for everyone. Also worth noting, there's a section here where they'll tell you news. A lot of times there's some really nice classes being offered on high performance computing. And the last thing you'll get is you'll see your username. You'll see it at some login node, and you will see you'll then see a $, and that's your cue that you can type in a command there. Okay, and we have a question coming in about R versus Python. So yeah, Marco already kind of answered on this one. R is basically a statistics analysis program with its own computing language called R. R has a lot of statistical packages already built. So if what you want to do is extremely statistics heavy, R tends to be a very good option. A lot of R scripts are not nearly as easily readable as Python. Python has an emphasis on scripts being extremely clean and easy to read. Python is really kind of a general purpose computing language. So for example, if I want to make something that sends out traffic over a network, Python's probably a much better option than R. If I want to run something that is trying to model a set of data I have versus a beta distribution for methylation analysis, R is probably a very good option because Python has the statistics packages, but R statistics packages tend to be much more mature. So typically the first command you run on the Hoffman cluster is QRSH to get yourself an interactive session. This will hang for a minute or two, and then you'll see it's your username at some node number. So that node number is a node you're actually on. And that tells you that you can now compute. So you can run stuff because you're no longer computing on a login node, you're computing on an interactive node. And you do that again by typing QRSH. All right, so there's two ways to run things on the Hoffman cluster. One of those ways is you start interactive node like I just showed you with QRSH. once you are in an interactive session, you can compute whatever you want on that computer as long as you don't go over your allotted resources. Alternatively, there's what's called the job queue. And if you use the job queue, you don't have to be connected. So to run things on the to run things interactively, you have to be actively connected, which can be really annoying because if you run something that takes a long time and you lose your internet connection, it'll actually kill your job in some cases. Alternatively, if you run it through the job queue, because that's being run entirely internal to the cluster will basically just figure out when it has the resources free for your job, and it'll run it then and there. And you don't have to sit there babysitting it, and it can just send you an email saying it started the job and then when it's ended the job. When you put in a QRSH, you can give it more options to tell it things like you want more data or you want more time. Getting data on and off the Hoffman cluster, you'll do that through CyberDuck or through another file transfer program. If you're using Linux, for example, you'll probably do this through via something like FileZilla. And this will actually be an interesting part because I'll help people set up CyberDuck, but usually to help people set up CyberDuck, I actually set up a student, although in this case, I obviously don't have that option. So I'm going to skip this part for now. All right, so I'm going to talk a little bit about sequencing file formats. And once I get through sequencing file formats, that's typically where I break the first day of class. So read analysis, what kind of stuff do you get? Again, you're going to get a bunch of files. So in this case, I'm showing QSeqs. More likely, you're going to get actually fast queues, but I want to talk about QSeq because people will occasionally get that from certain cores on campus. So the file name itself has information. The first part's telling you the lane. The second part's telling you the redirection. The third part is telling you the file number, the tile number for it. So in QSeq format, you're going to see that you get a few different things. So here we have field 1. So it's going to tell you the machine name, the run number, the lane number, the tile number. So machine, run number, lane number, tile number. It'll tell you the X coordinate of the spot. So the X coordinate of where it was observed, the Y coordinate of where it was observed. It'll tell you the index. In this case, there was no indexing, but if there is an index, it'll tell you the number of the index. It'll tell you the read number. So it'll be read number 1 for forward, read number 2 for reverse. So in this case, these are reverse reads. You'll have this long string of A's, T's, G's, and C's, and that's your DNA sequence. And then you'll have this string of what appear to be just a bunch of random characters. This is actually your quality score. And I'm going to go in depth into how to understand what this means. And then the last thing you'll have is, did the read pass Illumina's quality filtering? If it gets a 0, it didn't pass quality filtering. If it gets a 1, it did. Some people think Illumina's quality filtering is not stringent enough. Other people think it's too stringent and they ignore it. That I leave to the individual users. So that was QSeq format. And QSeq format, as you notice, everything is on a single line. So in QSeq, everything here from the first value, which is the machine name, all the way out to pass or fail quality filter, it's all in a single line. This does make it easier to read and write the files, but it doesn't make it easier to understand for a human. FASTQ format uses 4 lines per read. So the first line is going to have what I call the housekeeping data. Things like, did the read pass quality filter? Where did the read come from? Where was the read read on the flow cell? That kind of thing. Second line is going to be your sequence. Third line is going to start with a plus, and occasionally you'll have stuff on here, but those are just typically notes. Fourth line is always going to be your quality string, and again, I'll go into how to understand these. So FASTQ format uses 4 lines per read. I think this is actually why it caught on, is if you look at this, you realize that FASTQs are much easier to read in terms of being able to quickly pick out your sequence and not have too much crap on one line. So with multiplexing, I'm actually going to skip teaching people how to demultiplex because what I found in general is that the cores tend to do the demultiplexing for people. If you do find that you have to demultiplex, and so the people who typically do get multiplex data back usually already have a script set up for doing demultiplexing in their lab. If you find that you have to demultiplex, but you don't have a script available for it, let me know. I have some scripts I can lend you that work really well in the Hoffman cluster and tend to be pretty fast. So again, with FASTQ format, a FASTQ is going to devote 4 lines to each read. FASTQ format is the most commonly used by aligners and quality control programs. Basically, I don't see any programs these days using QSeq, so pretty much everything is going to expect FASTQ. I'll do this for now. All right, so again, with raw data format, this is going to help you start understanding what the quality scores mean. So quality scores are trying to get a lot of information into a little bit of space. But the quality score is basically trying to tell you for each base in your sequence, how confident was your sequencer in the read? So this is the original quality scoring scheme here, Sanger score. And here we see it's Fred 33, and I'll explain what that means in a little bit. We see here that characters for Sanger sequencing quality scores, and Sanger sequencing, by the way, is now the standard for even Illumina reads. Illumina uses the same Sanger standard. We see it goes from the exclamation point all the way out to the capital I. So quick show of hands, who out there knows what ASCII is? ASCII. Anybody? Okay, I got 2 people raising their hands. That's usually about what I expect. Okay, 3 people. All right, the basic thing about computers is computers don't really know text and they really don't know letters. Computers know numbers. And in fact, computers only really know 2 numbers. They know 0 and 1. If you string together enough zeros and ones, you can actually make a binary number, and computers can count above 1, which is kind of useful. But computers really don't know letters. So the only way a computer knows how to store text or store text information is it needs a way of mapping letters and numbers. And fortunately, in this realm, there's a very long-standing standard called ASCII, which is essentially a map of which letter goes to which number and which number goes to which letter. This becomes useful because for sequencing, you have a single base and you want to assign it a quality score. So let's say I want to assign it a quality score of say 33. I can give it an exclamation point. Rather than using 2 characters to describe that 1 character, I now just have 1 character describing it that I can easily translate back. Now here we see 33, but it equals 0 because this is actually called base 33 for Sanger, where the base value is 33. So it's the ASCII value minus the base value. So this comes back to a quality score of 0. Quality score 0 basically means the sequencing machine had no idea what base was actually there. It read basically nothing interpretable. Now you'll see here that the quality scores all go through different areas, but we'll also see that for Sanger and for the latest Illumina, it's called base 33. And the reason for that is if you look at these 2, which are the base 33s, the exclamation point, which is ASCII character 33, is equal to 0. This one only starts off at 2 because really quality score 0 is kind of useless, but essentially the exclamation point would be equal to 0 if it went down to 0. For Celexa, this is called base 64, and we can see that here at 59, it's -5. So this goes -5, -4, -3, -2, -1, and then 0 is at 64. For Illumina 1.3, it goes 0 to 40, so we see 0 is equal to 64. For Lumina 1.5, it goes 3 to 40. It goes, I think that might be a typo. I think it might actually go 2 to 40, but basically for base 64, the at symbol is equivalent to, yeah, I think that's a typo up here, but the at symbol is equivalent to 0. So you can manually figure out what scoring scheme something uses just by looking at what the scores are. So for example, seeing this question mark means you might be in the Selexa range. Selexa uses a completely different formula, by the way, which is why it goes to -5. We won't worry too much about that here. Essentially, Selexa values at the high end are just about identical to their Fred values. At the low end, there's some big differences though. But by looking at the range of values you get, you can kind of figure out what you're dealing with. But essentially, all you really want to figure out is just, am I with a base 33 method or a base 64 method? If you happen to get back data with a base 64 method, which you won't get back new data with base 64 because practically nobody uses that anymore, the best thing to do is actually convert over to base 33, which is a very simple and straightforward calculation that a lot of different programs can do. But base 64 has fallen out of favor. Pretty much everything's base 33 these days. And again, just by looking at subsets of characters, you can get a pretty good idea of what your scoring scheme is. And once you kind of figure out that there's base 33 and base 64, really you just figure out the base value and you can kind of go from there. All right, so what's so important about quality score encoding? When you're doing sequencing, you get a lot of reads back. And each read has a lot of bases. And it's really useful to know when a machine calls a base, was it particularly confident in that base call, or does it think that the signal is kind of muddy and maybe it's only 10% confident? Some programs require you to know quality score encodings. Some programs can figure out the quality score encodings themselves. Some programs just assume you're using base 33. I haven't seen any programs that assume you're using base 64, except for programs whose purpose in life is converting base 64 to base 33. So again, make sure that your files are all one quality score encoding. There's a few different programs for this. FastQ Groomer is a pretty common one that you can use in Galaxy or on the command line. And that actually gets us to what we're going to start with tomorrow, which is we're going to run a program called FastQC on our data. So yeah, normally the first day runs all the way through the 3 hours, but it looks like actually I'm finished pretty early today. I think in part because we're not, the class doesn't have as much interactivity in this format. Although considering the future, I'm actually rebuilding some portions of the class, so I'm probably going to build in some more interactivity for online format. But anyway, I'll stick around for a few minutes and answer questions. So if people send questions in via the chat app, I'm happy to answer questions for a few minutes. Otherwise, once the questions run out, everybody's free to go and try and grab some lunch. And obviously, everybody, until tomorrow, stay healthy, stay hydrated. Okay, I have a request. If there's anything I can clarify that you should do for class tomorrow, if you haven't logged into the Hoffman cluster recently, and by recently I mean in the last month or so, or if you aren't confident you remember your password, try to log into the Hoffman cluster. Make sure you can log in through an SSH session, through something like PuTTY or through your terminal. If you find out tomorrow that you can't log into the Hoffman cluster, you're going to be stuck waiting for them to either reset your password or activate your account or whatever else you need. And you won't be able to participate along with the rest of the class in the material. Also, one other quick question. This actually is just related to a project I'm working on right now, but if anybody is working in a clinical lab doing coronavirus testing, shoot me a message, especially if you're doing qPCR-based testing. I have something that I'm working on a project right now you might be interested in. Do you need CyberDuck? You will need CyberDuck. However, we will actually download it during class tomorrow. I'll walk you through downloading a copy of CyberDuck. Another question. Can you get qualitative data from DNA-seq, like looking for gene variant frequencies in a mixed population? Yes, oh, okay, can you get quantitative? Are you referring specifically to maybe pooling a bunch of samples together and just looking at frequencies? Yes, if you have WinSCP, WinSCP is fine. FileZilla is also perfectly fine. I actually don't even, in my day-to-day work, I don't actually use CyberDuck myself. I like CyberDuck because it works on both Mac and PC. If you have Linux, FileZilla is probably the best option. And I know that the other course, the Linux course, teaches you FileZilla. All right, when you log into the Hoffman 2 cluster, should you put your ID and cluster password you got from registration? Yes, you should. Okay, example would be taking a mixed population bacteria with random mutations and comparing mutation frequency between different conditions. Actually, I can stop the share on this one. I can get back my screen. There we go. Yeah, that's something you can do pretty reasonably well. And in fact, that's actually a very good option. So you can look, for example, at just base to base, how much variation you see, and use that as a marker for mutation. Yeah, that's definitely for sure. And yeah, I noticed I had somebody in Elaine Shao's lab here, so I was guessing that was probably you. Are you doing, so Jonathan, are you doing 16S sequencing? All right, Jonathan, I unmuted your mic, you can talk.
Oh, sure. So mostly doing not so much 16x, but just doing like monocolonizations of mice and then looking at mutation frequencies between different treatments of the mice. So basically colonizing with one species of bacteria, but with different mutants or different groups of mutants between them.
Oh, okay. Let's see. Just give me 1 second. I have to answer a chat question quick.
Sure.
Yeah. Okay. So that makes sense. Yeah, I think that kind of this kind of sequencing is probably the one you want to do. Yeah, because you can error model Illumina sequencing pretty reliably. I wouldn't necessarily want to do a lot of nanopore because nanopore tends to make more single nucleotide mistakes.
Gotcha.
But yeah, you should be able to look at sort of changes in variability over some region, especially if you know of any mutation hotspots where selection isn't really an issue.
Okay, that makes sense. Yeah, I think we have some ideas of things we expected to vary or not vary. So that's kind of what we're hoping for, is starting off a little more targeted and then moving more broadly, or just trying to see what else we can actually get out of data we have.
Yeah, that makes sense.
Cool, thanks.
No problem. All right, are there any more questions? Okay, I have a request to explain quality scores one more time. So quality score is a function of how confident the machine is in a base call. So I can show, I'll actually show the formula for it again a little bit later, but the rule of thumb for looking at quality scores is they're going to be Fred formatted. So it's called a Fred score. And the rule of thumb to go by is a Fred score of 0 means the machine had 0% confidence in its base call there, which means it basically didn't call a base. In fact, usually you won't even see a base call there. You'll just see a dot or an N or something saying it didn't have anything. A Fred score of 10 indicates 90% confidence in the base call, or a 10% chance of mistake. A Fred score of 20 indicates a 99% confidence in the base call, or the machine thinks that there was a 1% chance it had the base call wrong. A Fred score of 30 is 99.9% confidence, and that indicates basically the machine thinks it had 1/1000th of a chance, a 1 in 1,000 chance of error. And a Fred score of 40 is 99.99% confidence, or the machine is basically saying that it thinks there was a 1 in 10,000 chance it made a mistake on the base call. Typically for single base calls, you won't find machines calling confidences above Fred score 40, with the exception of PacBio. So PacBio can actually read the same piece of DNA multiple times. And by reading the same piece of DNA multiple times, it'll then do a consensus value, which is basically like doing high depth on a single piece of DNA. And that can give you much higher confidence scores. So it'll read the same piece of DNA 5, 6, 7, 15 times, whatever. And it'll read the same base over and over again. And if it reads the same base confidently 15 times, you're going to get a confidence much better than just 1 in 10,000 chance of error, and it'll indicate that. So so in the case of PacBio, you'll sometimes get confidence scores. You'll sometimes get Fred scores of like 90, which are insanely high. Does that make sense? Okay, and I'll go over it again tomorrow because quality scores are important. As far as credits for 275, I don't actually handle those myself. I don't assign them. Those are typically handled by Eloy. I think the only requirement is he just has to, I think he just has to confirm that you attended. Okay, and Anana's asking, are you asking how it relates to the formats I showed today? Okay, so I'm going to unmute you a little bit if I can. There we go. All right, Anan, are you able to talk? Okay, you don't have a microphone, so unmuting you was kind of not helpful. Okay, anyway, as it relates to what I was showing today, so what you would notice is the length of the quality score string and the length of your DNA sequence are always going to be the same, because the first character in your quality string will connect back to the first base in your DNA sequence, second to second, third to third, and so on. So if you remember when I was talking about ASCII, the reason why base 33 is base 33, because 33 seems like a random number to start with, is ASCII characters 0 to 32 are called non-printing. And those characters include things like tabs, line breaks. A beep is actually an ASCII character. So just have your computer go beep. That's actually, I think, ASCII character number 7. So you can't really put those into a file because they don't show up. But starting with the exclamation point, which I think is, which is 33 and the lowest numbered ASCII value that prints, if I have a base that I have absolutely no confidence in, the corresponding character in the quality string will be an exclamation point. And that'll be an ASCII value of 33, which 33 minus base 33 equals 0, telling me 0% confidence. Yes, exactly. So what you're showing is exclamation mark equals Fred 0. Yes. For base 33, exclamation mark equals Fred 0 for that base. Or in other words, what the machine is telling you is it put a base there, although for what gets an exclamation mark, you'll probably see a base of either N or a dot, which is just a placeholder saying it doesn't know what it read there. It didn't even make it didn't even try and make a base call. So does that make sense now? Yeah. So Anand, does that make sense to you? Okay. And okay. So I have a question. In the knockout mice example, I mentioned the N equals 3 to 4 in groups. And yes, Fred1 would be ASCII 34. So that would be the next character up from an exclamation point, which I don't remember what that is. N of 3 to 4 is generally not good enough for most statistical analyses. Although again, it's going to depend very much on what you're looking at. So just an example, I used to do, I did my PhD work studying a protein that when it was missing, mice couldn't clear lipids from their blood. And typically these mice ran triglyceride values of about 5,000. A normal mouse typically runs triglyceride values of about 50. So I had very easy statistics on that because essentially there was no overlap between the groups. And in fact, there was a space between them of several logs. That being said, I still had way more than 3 to 4 mice per group because 3 to 4 mice is just pretty much never adequate. You can sometimes get a mouse that's sick in some other way and could be throwing things way off. So you really typically want to have, if something is kind of going to make your paper, like if something's going to be a major point you need to be absolutely confident on, I would probably want to have 10 or 15 mice showing the same pattern, and then do the statistics on that. Just because mice sometimes can get kind of noisy in terms of biological properties. I might be a little bit more willing to accept something with a smaller N if it was kind of supported by other data there, and if it wasn't a key finding. But for a really key finding, I would definitely, in fact, even for something supporting, an N of 3 is generally not what I would want. Okay, so Anand asking, Fred1 would be ASCII 34. Yes, if you're using a base 33 system, then a Fred score of 1 would be ASCII 34. Different platforms used to have different representations. So Solexa used to have its own thing, and Illumina used to go off Solexa, but now Illumina goes off of the Sanger, which is the base 33. And really the only real 2 Fred representations are base 33 and base 64. And base 33 is overwhelmingly the standard these days. I tell you about base 64 so you know that it exists, and you may encounter it, especially with old data, but really now just about everything represents in base 33. And in fact, if you do find yourself dealing with base 64 data, probably the first thing you want to do with it is flip it to base 33 because most programs now assume you're operating in base 33. All right, do I have any additional questions? All right, I have one coming in from Mo Ishihara. How does the sequencing machine detect reading confidence? So if you remember from that video I showed earlier on where I was showing all the different colored dots and the machine reading all the different fluorescence signals, a lot of the confidence comes from basically how clean and how clear the dot is and how clean and clear the fluorescence colored signal is. So for example, if it picks up just a bright, pure green signal, then it's going to be very confident that whatever base is indicated by green is the one that it just added. On the other hand, if it picks up kind of a wimpy red signal, or maybe it picks up a green signal, but it also notices that green signal has some signal in the red channel, it's going to be a lot less confident, and it's probably going to indicate that by saying it looks a little bit dirtier. So the confidence is essentially just a function of how clean it thought the signal was from the fluorescence of that base. So Mo, does that answer your question? Also, Mo, you're in Lily Wu's lab, right? Okay, please tell her I said hello. I rotated in that lab a long time ago. And then a question from Jijun Huang. So you also suggest to send samples for RNA-seq or other seq with biological replication n greater than 4. It's going to depend on what your budget allows. I think n of 4, if you're doing just a pilot study, I think n of 4 is perfectly acceptable. If I just want to see if there's even a chance that, if I have an idea already what's going on, yeah, I might start off with an n of like 4. It's possible that with an n of 4, you get such a big difference that you decide you don't need a lot more samples. But typically an n of 4, if a reviewer sees an n of 4 for something, unless there's a lot of sort of other lines of evidence supporting it, or a really astoundingly huge effect size, a reviewer looking at something with an n of 4 is probably going to start asking questions about if you're underpowered, or if you're just subject to random variation. All right, does anybody have a question? All right, if anybody has questions, chat now or forever hold your peace. All right, I don't see any more questions, so I am going to, so I will end the meeting, and I will see everybody bright and early tomorrow morning.